Welcome to ASM Live, the 2013 Internet talk show from the ASM General Meeting in Denver, Colorado. I'm your host today. I'm Stanley Malloy. I'm the Dean of the College of Sciences at San Diego State University, and I'm the chair of the editorial board for Microbe Magazine. We welcome you today to send any questions that you have if you're watching us through Twitter to hashtag ASM2013. And for all of the people in the audience, we ask you to please ask questions when you have questions. Just come to the microphone if you have any questions. We're here today with Jayne Rapier. Jayne comes from Hunter College of the City University of New York. She has a very interesting story that combines learning about a natural resistance process in nature and potential application of that process. Can you tell us a little bit about your story, Jayne?
Sure. So I work on a factor in our blood known as the good cholesterol. And so the good cholesterol is responsible for taking fat that accumulates in our arteries to the liver and then delivering it. But aside from doing that, we have something that I like to call the very good cholesterol, which contains a key protein called apolipoprotein L1 that is very good at killing many things in our bodies. And it was discovered for killing African trypanosomes. So the way this particle works is it circulates in our blood freely, and all of us are born with it, so it's part of our innate immunity. And if you get infected by an African trypanosome, the parasite eats this ball of fat thinking that it's getting lipid in order to grow. And in fact, in there is a little Trojan horse, this apolipoprotein L1 that we make, and it very effectively forms holes in the membrane of the parasite and kills it.
So now we have to worry about bad cholesterol, good cholesterol, and very good cholesterol.
There you go, we do, exactly.
So how was this discovered?
Originally?
Yes.
So people knew that there were certain parasites in Africa that infected human beings, and there's two varieties, one from the west side of Africa called Gambiense, and one from the east side of the Africa called Rhodesiense. And then there are many other parasites that infect all of the animals that are prevalent in Africa, including livestock. And we are able to kill all of those other parasites. And so people started studying what was it that we had that enabled us to kill all of those other parasites. And slowly they found out it was the good cholesterol. And then with time, we realized there was very special proteins attached to that good cholesterol that allowed the factor to work.
And I expect that there must be some animals in Africa that have developed this resistance as well?
Yes. So as it happens, not just humans have this great protein, but so do gorillas. And they are very close to us in evolution. But surprisingly, the chimpanzee and the bonobo do not have this protein at all. They don't have this factor, and they are highly susceptible to the disease. So we don't understand why they didn't keep it, and the gorilla did. Then on top of that, there are 3 other primates that we're aware of in Africa that have this factor, and that would be a sooty mangabe, a mandrill, and the baboons. And they all have a factor that is actually far better than ours, because they can all kill human infective parasites, and we cannot.
Is it clear why their factor is better than the human factor?
So we've been working on that for some time, and we have I have some answers for you. So their factor is very similar to ours. There's about 60% similarity. And what we did was we took the gene that encoded for that factor out of the baboon, and we put it into a mouse. And we challenged the mice with the parasites, and we asked the question, is this gene necessary and sufficient to protect the mice from the parasite? And indeed it was. So then we started to make chimerics. We mixed the human gene with the baboon gene to start finding out which piece was most important to allow the baboons to kill human infective parasites. And we narrowed it down to the very end of the C terminus, so that's one end of the gene. And in fact, as we worked longer and longer on it, we found all you needed was one change. You needed one amino acid, so that's a building block of proteins. You needed one amino acid change, and that would enable you to be completely resistant to human-infected parasites.
That's an amazing story. It's particularly amazing when you think about how a population would develop this resistance and how evolution would occur out in nature. When you talk about these parasites, is the resistance very broad against different parasites, or are there specific parasites that show this effect?
So there are specific parasites that show this effect. And so what we found is that one of the human-infected parasites has made what we call a parasite immunity protein, and we've called it the serum resistance-associated protein because it associates with resistance to serum. And it turns out that the parasite made this protein new by modifying one of its own genes that already existed. And it's now able to infect a human being because when the particle, the good HDL, is taken up by the parasite, they meet each other inside, and the parasite immunity protein binds to our good cholesterol and prevents it from forming pores in the membrane. So we know very well how that parasite has evolved to be resistant to our own protection. Mm-hmm. There's the other parasite in the West Coast. We don't quite know how that's done. Its protection. We are beginning to understand that it's changed a receptor on its surface that it uses to accumulate the good cholesterol. And it's made it much worse in that it doesn't take up so much. As a consequence of that, it doesn't grow quite as fast because you need cholesterol to grow, and so they grow very, very slowly, and they don't appear to be getting killed at all by our immunity factor. So that's another area of research that we have to study.
Now, the trypanosomes you're talking about are in Africa. What happens if you look at this really important trypanosome in South America, Trypanosoma brucei, that causes Chagas disease? Do you see a similar kind of effect there? Do you know?
No, so we studied that in mice because we do fortunately have an animal model in mice, and so we can infect mice with the South American Trypanosome Lytic Factor. And what happens there is there's no effect whatsoever of this factor on that parasite, and it's likely because the African trypanosome swims around in blood, and it grows in blood, and it never goes and hides inside of a cell. And so it's always being bathed in our Trypanosome Lytic Factor, and it's always eating it. The brucei invades the cell and then breaks out of the little vacuole that it invades and grows in the cytoplasm of that cell. And so it's completely protected from our good cholesterol because it's hiding inside of a cell. And so there's no accessibility to the parasite.
Mm-hmm. Another very important parasite is Leishmania. And how about Leishmania? How does it respond to these situations? Because it's also an intracellular parasite.
Right. So Leishmania is, as it turns out, susceptible to our good cholesterol. And so I'm beginning to think that perhaps we shouldn't call it Trypanosome Lytic Factor anymore, and we should actually think of it as a broader antimicrobial lipoprotein. And so the way Leishmania infect a cell is they're taken up into a vacuole, and that's where they like to grow. And they like to grow in there in a very acidified space. And they do very well inside macrophages, which are the cells that are supposed to eat them and destroy them, but in fact they've taken them over to use them as their home. And it turns out that these macrophages will also take up our good cholesterol. And the parasite and the good cholesterol meet each other inside this vacuole, and the good cholesterol is superb at killing the Leishmania. However, unlike African trypanosomes where the immunity is sterile, and by that I mean you get complete killing of the parasite, with Leishmania there's a small pool that remain resistant, and we're trying to understand why that is. And so the immunity that we're given by having this good cholesterol is that we have a lesion that is perhaps the size of a penny as opposed to the size of a $1 note. And so the innate immunity keeps the number of parasites down so that we don't get a huge infection. And then eventually your adaptive immunity kicks in and takes care of it.
It's a really fascinating story of host specificity in recognition of different microbes. I'd like to remind the audience that we'd love to have questions. If you have any questions, please come to the microphone to ask your questions. And for anyone listening online, you can send questions to hashtag ASM2013. So it's an interesting story when you start to begin to say, okay, given what you know now, can you predict ways that you might think about using this approach to develop resistance? Right? So, for example, could you move these genes around and maybe alter the resistance in a cow?
Yes, so that is definitely one of our plans. So in the central area of Africa, cows cannot be grown isn't the appropriate word. They just don't exist too well because it's the tsetse fly belt. And this tsetse fly belt is about the size of the United States of America, so it's a very big area of land. And cows are supremely susceptible to this disease. And so when the tsetse flies bite them and give them the parasite, they get a wasting disease and they die. And so our idea has been to create a transgenic cow. And by that I mean take the gene out of the baboon, because that's the gene that kills all of the parasites, and give it to the cow. And so we're in process of doing that right now. We're working with a group in Nairobi, in the International Livestock Research Institute in Kenya, and with a group in Michigan State in the United States. And we're both groups are trying to get this gene into the cow so then we can then test and see if the cows are indeed resistant, just like the mice, to this disease.
So you don't have cows in downtown New York City?
We don't have cows in downtown New York City. No, they are very happy in Michigan State with lots of space, and also in Africa with lots of space.
This is really exciting because will have such an important impact on the health of the human population in that region of Africa where there's desperate need for better nutrition, especially protein.
Exactly, and it's not just for eating. So right now in Africa, because they can't have the cows in those areas, they actually plow their fields by hand. So 90% of the land is tilled by human beings working away with their hands. And so if we could just let them have one cow that was strong and wasn't susceptible to the disease, then they could plow a lot of their fields and actually have food, not just meat, but actually grow their own crops in a much more efficient manner.
So this is a very, very important international problem that has a lot of potential ramifications. We have a question now from the audience.
Hey guys, I wanted to ask if you would predict or might know if your factor might have any antiviral.
So indeed. So likely it does. There is a family of these genes. The one that is secreted and bound to the good cholesterol is not the only one. So remember, the pore-forming protein is called apolipoprotein L1. Well, we happen to have 6, and we don't know what they do. And recent work is shown, and none of the others are secreted. They all remain inside the cell. And so we presume they have some kind of antimicrobial activity, but we don't know what. And so there's new data coming out showing that one of them, number 6, appears to have robust antiviral activity. So it's very exciting because not only do we have a factor that's secreted into our blood that is antimicrobial, but we're also making ones that are in our cytoplasm to take care of things that infect our cytoplasm.
That's very interesting. Now, how was that discovered? That's a very different story than the cattle in Africa.
So it was done with a genetic screen, and there was absolutely no bias to the screen whatsoever. And the idea was that you take a cell and you infect it with a virus, and it infects very well. And then you take that cell and you introduce genes from from our entire genome, one by one, in the individual plasmids. And then you find out then you infect the virus and you ask, does the virus infect just as well or less well? And then you sort out, then you'll find that there are some genes that are in our genome that will prevent the growth of viruses. So then you select those genes and you look and see what are they. So you sequence them. And then you test, is this really the gene? So you put it back in and you find that it is indeed restricted for viral growth. And then all the hard work begins to sort out what's going on biochemically inside of the cell that's preventing that virus from growing.
When you say antiviral, is this broadly antiviral, specific types of viruses?
It's early days, but the APOL6 gene, it likes to go for what we call RNA viruses, viruses that need to grow inside of the cytoplasm and don't rely on the host membrane in order to envelope itself. So for example, HIV has an envelope because it takes the host membrane and uses it to coat itself. And then there are other viruses that do not. They just have what's called capsids. And so it's the viruses that make capsids that seem to be the susceptible guys.
Now, in this case, the mechanism must be considerably different than that of the parasites.
We assume so because viruses don't exactly have membranes, and so you can't exactly poke a hole in it and make it susceptible. And so we're assuming it's an entirely different mechanism of action, but this is to be worked out.
So what about bacteria?
So we've started testing bacteria, and we find that they are quite susceptible to our good cholesterol. So that would be apolipoprotein L1. I don't know anything about intracellular bacteria that grow in the cytoplasm of cells. That is yet to be discovered for all the other 6 genes, if any of them control bacteria inside cells. Certainly, the bacteria that we've looked at that can kill, that are killed by our good cholesterol, is the same principle as the Leishmania. They have to grow inside a phagosome that is acidified, and so that the cholesterol can be activated. And so we've looked at Salmonella, we've looked at Bacillus anthracis, and we've looked at Mycobacterium tuberculosis. And whilst we find exquisite sensitivity in a test tube, we're not finding such a robust sensitivity in vivo when we do our mouse models. So we haven't completely given up. We may be doing the wrong kind of thing, but certainly we're getting a hint that this factor can also kill bacteria.
So again, we have another question from the audience.
Hi. This is a great example, I think, of a fascinating basic discovery that has a translatable application. And I know that transgenic cows have been made previously, so there seems a high likelihood that this whole project could actually succeed. And I was just wondering, from your expert perspective, what do you think the toughest step of the whole project actually is?
So this gene, there's 2 things. First of all, getting the gene into the cow is proved quite tricky. So we've been able to make mice that are transgenic, i.e. they contain the baboon gene. And initially, people tried just taking what we call the cDNA. So you just take the sequence that precisely encodes for the protein, and you put it into the genome of your host animal randomly. You never know where it's going to land, and you give it a special kind of controller called a promoter that turns this gene on. And it turns it on, you can make it cell-specific or not, but it just turns it on. Those mice were always dead. And it's not surprising because it's a gene that makes holes in membranes, and if you don't regulate it, then the minute it's on, it's going to make holes in the membranes of the cell in which it's made, and it will die, and you'll kill your host. So to be a little bit more tricky, what we did was we took the whole locus out of the baboon. So this is a big piece of DNA. We took a huge piece out, and we looked at all the different marks along this piece of DNA to see where's the controller. Where's the piece that maybe regulates? And we took all of that and we put it into the mouse. And that worked. So we got viable animals out that are presumably making the protein. They're in quarantine right now, so I can't tell you. I wish I could. But we're using exactly that same piece of DNA to go into the cow. And we're also making it targeted. And so by that I mean we're putting it into a very precise place in the genome because we want it to be always on and controlled by the entire elements that are going with it. And we don't want the host genome to shut it down. So it's been a bit tricky. It's taken a lot of molecular biology, but I think in the end, it'll be worth it. Then the second problem is getting people to accept transgenic animals in a country where they're currently a little frightened of that kind of technology. And we're hoping going forward that it's simply giving the farmers choice. And right now, their choice is to use a compound that's like ethidium bromide. It's called homidium bromide. And it's quite toxic. It intercalates into the DNA. And that's what they use to treat their animals. And they use it once a month. So we're hoping they can have the choice of having a transgenic animal that could be sustainably resistant always, because the baboons have been around for 9 million years, and they still have not become susceptible to this parasite.
Presumably, you could modify the cattle so that they would also prevent transmission of human disease as well, correct?
That's absolutely true. So the cows act as reservoirs for the human disease, and they are in fact instrumental in propagating epidemics in Africa because human beings live in close proximity to the cattle. And these parasites are zoonotic, so they will grow in humans as equally as well as they will in the livestock. So even though they're throughout Africa in all of the wildlife, people don't hang around lions too much. So the transmission between those species, between lions and humans, doesn't occur as much as it does between cows and humans. So yes, we'll provide cows, we'll reduce transmission of the human infective disease, and so all around it should be a good win-win.
You know, this is an interesting story. Coming back to the focus on Africa, a lot of the investments of major international pharmaceutical companies in developing therapeutics for disease are focused on first world disease, if you will, developed countries, because that's where the resources are to reimburse the cost it takes to develop those processes. What you're working on is something that has a very tight focus here in a part of the world that does not have a lot of money that could ultimately pay for the therapeutics. So how do you support this kind of research?
So we're being supported by the National Science Foundation and the Bill and Melinda Gates Foundation. And they gave us a decent pot of money to at least pilot this study and see if we could create the cow. Our aim going forward would be to have actually a bull and to offer sperm to farmers through artificial insemination, which is a technique that's used frequently in Africa already. And I'm hoping, because of the Bill and Melinda Gates involvement, that this would be given freely, because after all, they do say every life is created equal.
I think it's very exciting to be working on something where you really have the chance in your lifetime to see a profound impact on many, many people's lives. So I hope that that happens sooner than later. So, but you know, I'd like to come back to this cholesterol. So often when we talk about good cholesterol and bad cholesterol relative to heart disease, we talk about modifications by diet. So is there a dietary impact on the relative abundance of very good cholesterol?
So all I can tell you, I mean, I honestly don't know. Maybe as more and more people get interested in this, we'll be able to answer that question. But I can tell you when our donors, we beg our donors not to eat before they donate, And you know, when you go to blood centers, they always want you to eat before you donate. And we always have the nurses who work in the blood donor centers collect the blood for us. So it's all with approval and everything. And so they turn up and the nurses go, have you eaten? And our donors go, no. And we say, no, don't tell them that. Because what we see is, if they've eaten before they donate, then the amount of very good cholesterol we get is severely reduced. But if we get them fasted, then we get really good yields. So we've just noticed in our own little world that there appears to be a difference, but it would have to be, you know, a proper study with lots of clinicians involved to say anything for real.
I think that thinking about such dietary intervention would also be very intriguing. I guess you would expect probably it would negatively impact people from developed countries more than from developing countries in terms of fat and cholesterol intake in your diet.
No, I mean, people in developing countries tend to be skinnier and slimmer than us, and so I would say they probably have really good, very good cholesterol. And in terms of the pore-forming protein, there's been some data now coming out showing that it gets increased in the face of infection. So it has some elements in its that control its expression that promote more protein being made when the body senses infection. So I think they're probably better off than we are. And in fact, there are variants of this protein in Africa that are actually pretty good at killing African trypanosomes that infect human beings. And so there appears to be a selection of a slight change in the sequence that gives these African people an advantage over any European.
So is that another single amino acid change somewhere else in the protein?
So there's two different variants that are running around Africa that we're aware of at this moment in time. And one of them is a deletion. 2 amino acids were deleted. And what that does is it brings an amino acid called a lysine into exactly the same position as we see in the baboon. So those people are pretty well protected. Partially, not 100%, but partially protected. And then there's another amino acid substitution a little further up in the sequence, and that also gives protection, but we can't quite understand why. We're still working on that one. The downside of having these genes is if you're homozygous, which means if you have 2 copies and they're identical, then by the time you're 30 or 40 years old, you get kidney disease, and you get quite sick. But in terms of evolution, it's not such a bad thing, because by the time you've reached that age, you've had your children, you've survived the disease that's endemic in that area, and your race continues. And if you're a heterozygote, it's perfect, because you're protected from the parasite, but you don't get the kidney disease.
Not so different from the story with sickle cell anemia.
Precisely.
Right? So
Yeah.
I think that that's a really important point. If the disease is bad enough, you'll have a heterozygote advantage there.
Right,
So,
right.
absolutely. So this has been really very interesting discussion. Thank you so much. It's a great story. It brings in our discussions about good and bad cholesterol. It talks about the importance of diseases in other parts of the world, that neglected diseases, the potential that this is going to have applications to viruses as well as parasites makes it really relevant to the first world, and potential therapeutic intervention that could happen in our lifetimes. Very exciting. It's so nice talking to you, Jayne. Thank you so much.
Thank you. Bye.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this.
Anybody who has access to this facility first has to go through an R scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.